effort, because we shall be contributing our ideas, to draw a reasonable conclusion on the role of certain features in drug design. Once again, you are welcome. Quickly before we start, I'm going to call on one of our team members to give us a brief citation of our speaker. So, Divine, I don't know if you're here with us.
Yes, I'm here.
All right, so we are going to take 5 minutes for citation reading while I call on our speaker. Thank you.
Good evening, all. I'll be taking the citation of our speaker. Okay, Dr. V.O. Imeje, PhD. Dr. Vincent Ogenerubo Imeje, PhD, is a distinguished pharmacist, lecturer, and researcher in the Department of Pharmaceutical Chemistry, Faculty of Pharmacy, University of Benin, Uniben, Benin City, Nigeria. With a career that breeds rigorous academic training, international research experience, and significant contributions to medicinal and natural products chemistry, Dr. Imeje has established himself as a leading scientist in pharmaceutical and natural product research within Nigeria and beyond. He began his academic journey with a Bachelor of Pharmacy (BPharm) degree from the University of Benin, laying the foundation for his lifelong commitment to pharmacy and medicinal chemistry. Following this, he pursued a Master of Science (MSc) in pharmaceutical chemistry at the same institution. Driven by a desire to deepen his understanding of drug discovery and natural product chemistry, he later enrolled for a Doctor of Philosophy (PhD) in pharmaceutical chemistry at the University of Benin. His doctoral training equipped him with advanced expertise in phytochemical analysis, compound isolation, structural elucidation, and bioactivity screening, skills that have shaped his research identity. Since 2016, Dr. Imeje has been a dedicated lecturer in pharmaceutical chemistry at the University of Benin, teaching at both undergraduate and postgraduate levels. His courses emphasize core aspects of medicinal chemistry, pharmacognosy, and drug design, equipping students with the scientific knowledge and practical skills necessary for modern pharmaceutical research. In recognition of his leadership and administrative capabilities, he is the head of Department Pharmaceutical Chemistry, University of Benin. Dr. Imeje's research has taken him beyond Nigeria's borders, exposing him to diverse scientific environments and international collaboration. He has engaged in research activities at the University of Rostock, Germany, a center known for strong programs in natural products chemistry and pharmaceutical sciences. Additionally, he has served as a visiting scholar at the National Center for Natural Products Research, University of Mississippi, USA, an institution with a global reputation for excellence in natural product research. These international experiences have enriched his research perspectives, expanded his professional network, and enhanced the global visibility of his work. Over the course of his career, Dr. Imeje has authored and co-authored more than 20 peer-reviewed scientific articles, many of which appear in reputable journals, indexed nationally and internationally. His publications encompass a wide range of topics, including anti-plasmodial activity of plant extracts, antimicrobial screening of isolated compounds, antioxidant profiling, and in vivo anti-malaria evaluations. A significant portion of his research portfolio is dedicated to exploring anti-malaria and antimicrobial properties of local botanicals, reflecting his commitment to addressing public health challenges that disproportionately affect sub-Saharan African populations. His work also includes in silico studies such as molecular docking and pharmacokinetic modeling, which help to predict how identified compounds interact with biological targets and their potential drug likeness. By integrating traditional wet lab experimentation with computational analysis, Dr. Imege exemplifies a contemporary multidisciplinary approach to pharmaceutical research. Dr. Imeji's contributions have been recognized through invitations to serve as a reviewer for several peer-reviewed journals, where he provides expert evaluation of manuscripts in areas such as phytochemistry, pharmacology, and natural product research. He is also actively involved in professional networks that promote pharmaceutical research and the application of science to improve health outcomes. His vision includes expanding the frontiers of natural products drug discovery, promoting the sustainable use of medicinal plant resources, and fostering collaborations that link African researchers with global scientific communities. Dr. Imeji remains dedicated to research that not only adds to scientific understanding but also has tangible benefits for society, particularly in the development of accessible therapeutics for diseases that burden communities in Nigeria and across the world. Dr. Vincent Ogenerebo Imeje represents the emerging generation of African pharmaceutical scientists whose work is rooted in traditional medicinal knowledge, advanced by modern analytical and computational methods, and oriented towards solving pressing health challenges. His academic journey, from local training in Nigeria to international research engagements, highlights a career that reflects both depth and breadth in pharmaceutical sciences. Thank you.
Can we give our doc? Can we give our doc a clap emoji? Yeah, yeah. He at this very point in time, I will close my mouth for him to take over. So, ladies and gentlemen, join me as I welcome Dr. V.O. Image to take over the platform. Thank you very much, sir. He's a man I respect so much, but I will not speak much. Thank you so much.
Well,
All right.
thank you very much,
Sir, you are welcome.
Dr. Henry, and to all the listeners. Uh, I'm amazed really at the citation because I was wondering if that was me or maybe somebody else, you know. I don't know how you put that together. So, well, thank you very much. That is wonderful. And also, I want to appreciate you for your dedication to expanding, this area of knowledge to not only to Nigeria but around the whole world, because we have people from India, different places, from East Africa, South Africa, trying to undergo this training with us. So thank you very much for the platform. Thank you very much for your dedication. And also, I want to appreciate all our listeners. Uh, well, in Benin, we try to do the best we can to further knowledge. And I think that is why we're here. And also, that is why uh, this interaction really is going to be collaborative. We are going to discuss knowledge and how we can, deploy this knowledge in solving our problems, both social and, economic problems. And also I'm very happy also for Dr. Henry because most of the research that he's doing currently, they are translational, and that should actually be the focus of our research going forward in Nigeria and maybe elsewhere. Translational research that you can bring your research from the bush, as it were, to the lab and then to the shelf, and that is very good, way to start. And I can tell you also, current researchers in Nigeria, they are focusing in that area, uh, and that is very beautiful. Uh, Late last year, we had a public lecture uh, given by Professor Martins Emeje, which is currently the DG of the Nigerian Traditional Medicine Board. And it was really exposing because he actually showcased some of the products from his research at the center that have been patented, and currently almost about five products that he showcased in that lecture that have been patented and currently in the Nigerian market for use in the treatment and management of different diseases. And so this is also our area of focus in the Department of Pharmaceutical Chemistry, University of Benin. And currently, I can also tell you that recently we have just concluded a training or a research work in trying to encapsulate some Nigerian medicinal plants that are proven to be used in the management and treatment of malaria. And so hopefully before this year runs out, that product will be patented and released into the market. So that being said, I think I would like to share my little slide with you. Like Dr. Henry said, we will not waste much time in this webinar. It's not like a regular training that we have to spend maybe 2 hours to to grab the content of the training. And so I would like to share my slides. Uh, just a minute, please. I want to ask, is my slide visible, please?
Yes,
Yes, sir, it is.
Yes, it
Okay,
is.
Yes, sir, it is.
okay, thank you very much. So this is going to be a very brief one. It's a webinar, and hopefully Dr. Henry will be in the School of Computational Chemistry and Molecular Biology will be hosting series of webinars for this year and maybe the years to come. And there are people who are here now, who are also very vast in different areas of molecular biology and computational chemistry. And so my topic today is structure-based and ligand-based drug design. Okay, and of course, they have already gave my citation. And so feature-based drug design. So by definition, feature-based drug design uses specific molecular characteristics, what we call features of drugs and protein targets to computationally generate or identify effective new drug candidates. That's what feature-based drug design is all about. And so because it is feature-based, and so certain specific molecular characteristics of features of drugs and proteins are used to computationally generate or identify effective new drug candidates. And that is the whole beauty of, computational chemistry. Uh, there's so much, like yesterday in the training, so many databases to deploy, to use in computational drug discovery. And the beauty of it also, one thing that is very useful or beautiful in drug in computational chemistry is the use of computers that actually may not cost so much, you know, to to design a drug and then of course test its potential efficacy. Potential because you may need to validate the efficacy of your new drug using in vivo models, before you can go uh, to possible clinical studies. However, uh, what we are doing in Nigeria, we may not have to even, you know, go much to spend money in drug discovery, especially in synthesis, because we don't have, you know, the instruments, the equipments, and the power to synthesize most of these agents in our country. I was in a discussion with the DG NAVDAC last year, and okay, 2 years ago, and that was the issue. You know, DG NAVDAC and her group, they wanted to see how drugs in Nigeria can be synthesized, and the especially APIs, active pharmaceutical ingredients, to be synthesized in this country. So I told her that this is almost uh, not possible because currently in Nigeria we don't have one single functional NMR. And so what are you going to use in, you know, structure elucidation if you synthesize a compound? Even even mass spec is very difficult to obtain in our country, maybe about two places alone. But currently, as we speak, there's no single functional NMR for structure elucidation in this country. So how can you possibly synthesize? So using, you know, herbal products or medicinal compounds from plants and, you know, different organisms is one thing that we can use in Africa, in Nigeria in particular, to really help our populations. And so, feature-based drug design employs methods like pharmacophore modeling, that is identifying spatial arrangements of features like hydrogen bonds, donors, and acceptors, which one of the I think training that we had, those short trainings that Dr. Henry really introduced most of us to, and also uh, use methods like quantitative structure activity relationship QSAR to predict biological activities, toxicity, and metabolism. Sometimes it may combine with AI, artificial learning, artificial intelligence, and machine learning to virtually screen and optimize lead compounds. And so this is the beauty where machine learning actually comes into play, because you can screen libraries of compound, thousands of libraries of compound, you know, at a go. And then, of course, from there you can be able to identify maybe a lead compound that can go into drug development. So, ligand-based drug design and structure-based drug design uh, uh, are two approaches used in the field of drug discovery. So, so what a ligand-based drug design focuses on the interactions between small molecules, that is ligands, and their target proteins. The interaction between drug molecules, small molecules, ligands, and their target proteins. That is what ligand-based drug design means. Using computational method, of course, to predict the binding affinity and selectivity of potential drug candidates. So that is basically what ligand-based drug design focuses on. And then structure-based drug design, on the other hand, involves the analysis of the three-dimensional structure of the target protein to design molecules that can bind to specific binding sites in the target protein and modulate their activity. So this is basically one, it leans towards the drug molecule itself, the interaction between the drug molecule and the target protein. And then structure-based actually is it leads towards the 3D structure of the protein. So you study the 3D structure of the protein to design molecules that can bind to specific targets in that protein. And of course, the binding modulates activity of the protein or maybe activity of an organism. Of course, this organism also they have protein targets that you can actually target. All right. So ligand-based drug design relies more on experimental data and known ligands. So you have to know the ligands, and then of course you have to rely on the data that have been curated from experiments in using this approach of drug discovery. Structure-based drug design offers a more rational and structure-guided approach to drug discovery because you're actually working from the protein, and then of course you know the targets in the proteins, and then use them to design drug molecules that can effectively bind to such protein targets, and of course cause a therapeutic effect. Both methods generally are good, whether you are using structure-based drug design or ligand-based drug design. They are good. They have their strengths and also their limitations. So, but again, they can be used in combination to optimize the design of new drugs. So, and that is the goal of any scientist who's working in the field of drug discovery. You want to optimize the propensity of the new drug or the synthesized drug or the discovered drug to be very potent without causing much side effects. Now, this is a little table that shows comparison of both concepts. Okay, and so we have the attributes and then the ligand-based drug design and then structure-based drug design. So target identification, okay, this is based on ligand receptor interaction. So you see a ligand with your drug molecule interacting with a receptor site in the protein. Okay, while structure-based drug design is based on protein structure. So you're actually investigating the structure of the protein and design suitable drugs that can, bind to the target site. And then the approach focuses on ligand properties in the ligand-based drug design. It focuses on ligand properties, while the structure-based drug design focuses on protein structure. So you are actually harnessing the knowledge of the protein structure in your drug discovery. And then flexibility in ligand-based drug design can accommodate flexible ligands, okay, flexible ligands. And that is, I think Dr. Henry was talking about in our first lecture, I suppose, when somebody was asking question on molecular dynamics. Now, most of the computational studies that we do without MD simulation is actually a rigid doctrine we are doing, really. Okay, but when you do MD simulation, there's this flexibility that the protein is in its natural, state, like as in the body. So there's this flexibility. The protein is dynamic. It's not just stable. And so the drug, if that drug can bind to the protein and remain stable for a period of time to elicit its pharmacological activity, then of course you can say that yes, this drug is a potential candidate, because most drugs may not be able to be stable long enough to elicit pharmacological activity. And so in ligand-based drug design, the molecules that can accommodate flexible ligands, okay, but structure-based drug design may not account for ligand flexibility, okay. Computational tools that can be used in ligand-based drug design include QSAR, okay, quantitative structural activity relationship, and pharmacophore modeling. And then pharmacophore modeling generally, is very useful in in drug design, drug studies. Okay. Then in structure-based drug design, molecular docking, molecular dynamics are very, very, useful because you have to establish that this, like I said before, that the drug binds to the target site and remain there long enough in such stability for it to elicit its uh, activity or its action. So drug discovery process also can be faster in ligand-based drug design and less expensive. Okay, and also, but for structure-based drug design may require more time and resources. If you recall what Dr. Harry was telling us a few last week, I suppose you need to run calculations in your molecular dynamics simulations, and we don't have these supercomputers to do such calculations. Okay, yeah. There are some calculations that will crash your small laptop down, in no time. So there are calculations that you can run for 3 weeks and going and calculate the thing is constantly running. We don't even have the power to sustain, stable power for you to run such calculations. We don't have, supercomputers and even big computers to run such calculations that you require in MD simulation. So it requires a lot of resources. There's a center that some of my colleagues that we use in Nigeria here, I think in the West, to run one compound, one ligand, MD simulation of ligand cost you to about 300,000 naira for one sample. And if you have about 10 molecules that you want to run, you can imagine what that will translate to. So it is a little bit more expensive and it requires time. All right. So this is a schematic representation of both structure-based drug design and ligand-based drug design. And so the early stage drug discovery is there you identify potential hits, you identify it. And then in your ligand base, you do your virtual screening, your 3D or 2D similarity based, okay, and also activity affinity prediction using your 2D, 3D QSAR. And then in structure base, you also do molecular docking, ensemble docking, free energy perturbation, okay, and then of course, your integration strategies. You can do parallel hybrid scoring and sequential, LiDAR screening. You can do all this. So if you have to do parallel, studies, it becomes you have to do ligand-based vector screening and molecular docking. So from there, you can obtain results, your N% hits from both approaches, okay? If you use ligand-based virtual screening or molecular docking, you have to obtain heat compounds that you can, funnel through for prioritization and ranking. Okay, then in the hybrid also, you can also do two of these together, then do hybrid scoring, and you combine the individual ranks together to have your compound ranking and you prioritize your compound. Sequential also, you do your ligand-based virtual screening. So you look for fitted, and hits, okay, the compound that fits into best fits, and then of course heat compounds, and then you do your molecular docking with the fitted hits, and then of course you do ranking and your prioritization. Okay, ligand-based drug design approaches start from an expressive database of ligands with known biological activity. Now, if you were in the training of yesterday, Dr. Harry introduced us to different databases. One that was really functional, which I love, was the in, in, the India medicinal plants database, which impact, which was very, very good. I think I love that. Okay, and then of course we also deal with Dr. Dukes. And then we have this traditional Chinese medicine, database also, and then of course phytochemical literature database, and the last one. But of course, we also have uh, an assignment on those. But I can say that the coconut and the impact, they looks very good and very reliable too. And so, yeah, so you from an expressive database of ligands with known biological activity. So where similarity testing can lead development of therapeutic analogs or select a therapeutic pathway to be investigated, like it's a target, you know. But in SBDD, on the other hand, starts from the known three-dimensional structure of the protein target. So, and then commonly uses the molecular docking technique, analysis of ligand protein interaction, to identify the best ligand model to in vitro and clinical studies. So in this SBDD, you actually focus, like we said before, the three dimensional structure of the protein target. And of course, with your molecular docking simulation studies, you can now analyze, these compounds, and then from there, the best ligands goes into in vitro studies and of course clinical tests, which is of course clinical trials. Okay, and then this also is scheme, okay, of the whole process, which includes target selection. Okay, and then from databases you use Chem Spider or Zinc database, collect your compounds, and then of course you funnel them in and ligand enrichment, okay, by structure activity relationship to find, ligands that are very suitable and like they have pharmacophoric features that you can use for your evaluation for clinical studies. Okay, so this is just a pathway, screening by docking, use different, uh, softwares or tools, docking tools for that. And of course, you do your biological screening and confirmation using in vitro or in vivo studies. Now, let's talk a little bit about structure-based drug design. And this slide shows a workflow of structure-based drug design. So we were saying that in this we focus on the protein. So this drug target selection based on the protein structure, and then drug target structure evaluation. You evaluate the structure, and then you carry out your binding site identification. You need to do that. Of course, we also have different tools to do that. One of the most prominent one that we use here, we use Pymol, and we use your Bioga Discovery tools that you can use in the identification of your 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 target structure or your binding site rather. And then of course you carry out your docking, and then of course in your docking you can do blind docking if you choose to. You can do, site directed docking if you want to. So you docking and of course scoring. Scoring in this case will be your binding affinity that you have to be able to generate from your docking. And of course, you identify the lead compound, and then of course you optimize. Okay, and then of course you all the compounds you may have to optimize. The reason you have to optimize because you are looking for a suitable candidate that will meet the desired objective of your study. And then of course from optimization you go to your drug candidate. So you'll be able to get, like, of course, in our general normal, a drug discovery that could take billion or over $1 billion in bringing one product into the market. Of course, we don't have such money. And then, of course, it take many years, close to 15 years, you know. But if you go through the route of of computational chemistry will completely, reduce the time to which you can actually bring a drug to the market. So you have a drug candidate, and of course, you go further for different, uh, you have to bring in the design of the drug to be able to be used, maybe suitable design using pharmaceutical knowledge to design whether it's going to be injectables, it's going to be, oral suspension and different things like that that we can use. So a whole lot of things can go into it before your clinical trials and the phase one, phase two, phase three clinical trials, that can be done to bring a drug to the market. All right. So the core principles here in structure-based drug design. So the target structure is I need to focus on that. So you must obtain the precise radio structure of the disease-related protein. Okay, you must obtain that, and you can do that via X-ray crystallography. In fact, most of the protein structures that are curated in the PDB, they are actually they also went through X-ray crystallography, NMR studies, and cryo electron microscopy. They have to go through all that to make sure that the structure that is curated, submitted, is a perfect structure. And then binding site identification, you need to pinpoint the pocket or the site where a potential drug will bind. You need to identify that. Okay, and that is why sometimes the site directed docking, I think I love using that because it gives you a focus on exact binding pockets protein that you can dock your compound. uh, the computational modeling, okay, this you can deploy your software to virtually dock or fit small molecules into the binding site, reducing their orientation and stability. Okay, then iterative optimization. In this case, we refine the chemical structure of promising candidates through cycles of computational design, okay, synthesis and experimental testing, of course, which is QSA, to improve affinity and the specificity of your heat compound. So these are some of the core principles that you can use in your structure-based design. Now, key techniques and tools. One of the techniques is molecular dressing is prominent. Most all these areas of study, molecular docking is very prominent, which produce how molecules fit into the target's binding sites, you know, uh, fit into the target binding sites. Okay. One thing I'd like to say, like in one of the slides, we're talking about ligand flexibility. If the person can mute his phone or her phone, that'd be fine. So I was saying that, yeah, please can you mute your mic, please? So I was saying that in talking about ligand flexibility, okay, the reason is that the ability, when you talk of ligand flexibility, you're actually talking of the ability of a molecule to change the shape, okay, conformation to fit into the target. Okay, so the target is there, and that is why also is very good to carry out MD simulation, especially if you need to, write, publish that work, publish that work. So that gives you some measure, or maybe editors or maybe reviewers of measure that actually yes, your heat compound that you are publishing has the ability to fit, and remain, fitted into the binding site for such a time long enough for it to elicit its activity. And this is very crucial, ligand flexibility is very crucial for effective binding in drug design. So the ligands, they just move like amoeba, to fit into the target and lodge in the binding pocket. So it allows for adaptability to protein pockets for optimal interaction. So the ligands can actually, fit into that place, remain there long enough for, to elicit its activity. So the other technique or tools is homology modeling. Creates a protein structure when experimental data is unavailable. This is actually where you deploy your molecular homology modeling. When experimental data are not available, then you use this to create a protein structure and to ensure the rationality of model structure, you have to do checks on the stereochemistry. If you model a structure, you must do checks on the stereochemistry, whether this compound actually have the stereochemical features for it to be a compound and for it to be able to fit into the protein that eventually you will dock into. The energy profile. Generally, the energy profile should be low enough. Okay, the energy profile should be low enough such that the molecule can actually elicit its activity. Residue environment and structure similarity are also needed in homology modeling. Machine learning and AI. Okay, this is also a very new area, and thankfully, Dr. Henry has promised to 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 run a training on machine learning and AI. So, and this actually accelerate virtual screening. Yes, you like the assignment they are giving to us. So we are afraid most time when you have to uh, single-handedly download maybe about 150 ligands, and then also, sometimes 500 ligands and sometimes 1,000 ligands. But with machine learning, that and AI, that can just take you before you go to your bedroom and come back. Almost the experiment is completed. Okay, so that accelerates virtual screening, predict properties, and generate novel molecules. And that's the beautiful part of machine learning and AI. You are likely to get more novel molecules, to use for your studies and for your drug discovery studies. So that is very important also. And then also experimental methods can also be used, like we said before, X-ray crystallography, NMRO, cryo, EM. Of course, EM is electron microscopy. All those ones provide the fundamental structural data that we use for your studies or your drug discovery. Now, protein structure determination. Now, first of all, in the protein structure determination, you need to talk about the folding recognition, okay? And in folding recognition, you have what we call threading, okay? Now, threading is a computational method to predict a protein's 3D structure by aligning its amino acids sequence onto known protein fold templates. Now, mark this down. In this one, you align the amino acid sequence onto known protein fold templates, known protein, not unknown proteins, known protein. Okay, and then calculate the probabilities of 3D protein structures could be formed if probability of 3D protein structures uh, uh, could be formed from the given protein. You have to calculate that also using your fold recognition. And then also both the environment of residue interaction and protein surface area are considered in the threading protocol. Yes, this threading here, I think this is wrong. I think there should be TH before you have your arrow. So you may need to correct that. I think that this is recorded, but you can correct this. All right. So both then structure of the higher probability is recommended to structure for the structure to generate the protein model. Okay. So the structure with the highest probability is recommended to generate the structure of the protein model. Now, there's also in structure activity relationship, there's ab initio protein modeling. Now, ab initio calculations really, if you are conversant with your Schrodinger equation, especially for those that are chemistry, I mean, chemistry and pharmaceutical chemistry days, you may have come across Schrodinger equation. Actually, Schrodinger equation are derived from ab initio calculation. And so, and that of course lead to different, uh, uh, models that they are using today for computational studies. And so first in this artificial protein modeling. Now, so in this one, you're building the structure from foundation for fundamental biophysical principles. That's why I make reference to Schrodinger. Those are fundamental biophysical principles that were used in to make calculations, especially in energy minimization. And that is why you have to minimize your energy, even when you are using your pirates in docking. You have to minimize your energy for you to be able to dock, because if it's not minimized, then you're going to have such, huge, protein, and then of course energy, uh, 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 the energy barrier, for your ligand to overcome before the docking can take place. So you need to minimize the energy, and then of course you have also to do conformational search. And so you have to use different conformation to make sure that you have the best conformation, and also you have to do some scoring functions and use force feed. Different force fields can be deployed in ab initio protein modeling. Based on physical properties, principles rather, residue interaction center and lattice representation of a protein to, build the target. So ab initio protein modeling is based on physical principles, residue interaction, centers and lattice representation of a protein to build the target. And also you use when you deploy this, when you use this approach when other protocols fail to predict an unknown protein structure. So you use the ab initio protein modeling when other protocols fail to predict an unknown protein structure. And also, you use this when the identity and the accuracy given by this modeling is lower than others. Okay, so you have to deploy this ab initio protein modeling. Note, ab initio protein modeling only models only the amino acid sequence alone, without only amino acid sequence alone is used in ab initio protein modeling without relying on known templates. If you notice, we said ligand based, structure drug discovery is based, it relies on known protein templates, but here it does not rely on known protein templates. So you have to mold that ab initio. Ab initio actually means from the beginning, from scratch. All right. Then also in your structure-based uh, drug discovery, you have this hotspot prediction. So hotspot prediction, you have to, in the protein, there are hotspots, okay, that you have to predict. So one big issue in structure-based drug discovery is to determine ligand active site. It's a big problem, and so you may have to predict the hotspot in the active site, okay, before you can, use structure-based drug discovery. And then also you can determine the hotspot using X-ray crystallography. It is a very powerful tool in drug discovery, and also in structure elucidation. Also, you can use X-ray crystallography. And one of my colleague in Uniben, he did some work in Pakistan where he used X-ray crystallography. Also, I did one work in Germany also that I used X-ray crystallography to actually help me determine the structure of the compound isolated. Painfully, the compound was reported, though we were thinking it was a new compound, but we realized that after going through our site finder, we realized that the compound, somebody had reported the compound in 1976. And so that makes us very, very sad, you know. So X-ray crystallography is a very powerful tool that you can use in structure activity. Your structure-based drug design. Then also hotspot prediction, but not it is not possible for proteins that cannot be crystallized. You cannot use this for proteins that cannot be crystallized, and that is why most of the proteins in your PBB is they actually crystallize. They are crystallized structures of the proteins, and that is what you download in your download your PubChem. You download the crystal structures of the your protein. Now, so also several binding site determination methods have also been invented that can aid in hotspot prediction. Okay. All right. So another thing that you have to carry out also, if you look at the scheme that we presented above, is docking in your structure-based drug design docking. Now, of course, docking, almost everybody knows about docking. The docking practice the preferred orientation of one compound to a second when bound together to each other. So it predicts the orientation to form a stable complex, because the whole beauty of it is that there must be a stable complex that is formed. Okay, now docking you when used when other protocols fail to predict an unknown protein structure. Okay, now the identity and the accuracy given by this modeling is lower than others. That is docking. Okay, uh, the accuracy and the identity is lower compared to other methods. Okay, note we said before that. I think I've repeated this. I think I've done this before. Okay, so go to the next one. Yeah, so this is scheme also in the docking process. And this is our protein. This is the binding site in the protein, and these are ligand. And so we talk a ligand to the protein, okay? And then there's a ligand protein complex, which of course is shown here. So basically, it's a ligand plus protein. It gives you a complex, of course, and it is this complex that we now begin to analyze. Okay, we do our analysis based on the complex that we obtain from our binding of our ligand to the protein. All right. So there are popular docking software, okay, that can be used in our drug discovery, especially structure-based drug discovery. Now, one of them is Autodoc. Autodoc is very, very is prominent, very popular, okay, very efficient in docking small molecules or small ligands into a rigid 3D structure. Very Autodoc is very, very good. Then, of course, we have different ones, of course, these ones listed are based on the number of citations that, number of articles or work that have cited them. So we have Autodoc, we have Gold, we have Doc, we have Flex, we have Glide, FT Doc, and QSP. These are different, software that you can use for your docking. And you may have to study. There are different ones that are available out there. You can use Schrodinger suite, the rest of them, you can use that also for docking, you know. Then let's talk a little bit about ligand-based drug uh, dis- uh, oh, my, drug design. I think this is supposed to be about 1 hour. I think I have exceeded that. I'm going to be fast a little bit. So ligand-based drug design relies on ligand-based drug design, as I was saying, relies on knowledge of other molecules that bind to the biological target of interest. So in this case, it relies on the knowledge of other molecules that bind to the biological target of interest. These other molecules may be used to derive a pharmacophore model. Okay, we rely on the knowledge of other molecules that bind to the same target, and then from there we can use that to derive a pharmacophore model. Now, actively, a QSAR relationship in which a correlation between the calculated properties of molecules and the experimentally determined molecular activities may be derived. Okay, alternatively, you use a QSAR relationship. Then QSAR may be used to predict the activity of new analogs. Okay, of course, uh, Dr. Harry or Siago, right? I think Dr. Siago we will give us a training on QSAR. All right. And so quantitative structure activity relationship, QSAR employs statistics, analytical tools to investigate the relationship between the structure of ligands and their corresponding effects. So basically, that's what and this is all about. And of course, from human medicinal chemistry, we know this, what we call structure activity relationship of a drug molecule of your compounds. Okay, so you investigate the relationship between the structure of ligands and their corresponding effects. Okay, then also relies on mathematical models are built based on the structural parameters to describe the relationship. Then earlier 2D Q star for 3D have been adopted. So earlier, before this time, we were using 2D QSAR, but now we have 3D QSAR also. And that is why, if you notice, when you are dilating your protein, or I mean your ligand, yes, your protein from your protein data bank, we're often told that if you have the 3D conformer, it's better for you to download using 3D conformer, but except when the 3D conformer is not available, you can now use your 2D conformer, and that is very important. The 3D QSAR methodologies include COFA, COMSIA. Now, most of these ones, actually, they are available and they can be used. However, I'm going to talk just today based on COMSIA, okay? Now, it's a comparative molecular similarity index analysis. The COMSIA, you use this to comparatively, check the similarity index of your ligand and the protein. Okay, then includes also more additional feed properties. Okay, you check steric effects, okay, and then electrostatic effects, and then you have hydrophobic, hydrogen bond donor, hydrogen bond acceptors, and then of course, if you notice, these features are also the ones that you describe in your 2D, analysis of your dot compound. Okay, these are the things you actually discuss, and the hydrophobicity, hydrogen bond donor, hydrogen bond acceptors, and that also you can use in your structure activity relationship. So can offer comes here this platform can offer a more accurate structure activity relationship than COFA. Okay, and so and then continuous structure activity relationship QSAR, like we said before, this is just a pictorial, confer and COMSIA. This is our ligand, okay, and so you have spots on the ligands that are very, you know, so this ligand is docked into the protein, okay, and so this is just a pictorial of the confer and the uh, COMSIA. All right. Now, in conclusion, I would like to say that molecular simulation has a vital role in drug design and computer-aided drug discovery. Very strong vital role, molecular simulation, and that is why you may need to, carry out that, especially if you are not just studying for possibly production or maybe formulation into the market. Okay, for most articles that you need to publish, you may have to do MD simulation. That's very vital in drug design because you have to know if the molecule is stable enough, the binding, the protein, they are stable long enough to elicit the activity. But it's also a molecular, computational study is a very fast, efficient, and inexpensive tool to discover new possible ligands against a molecular target. Very fast, very reliable also. Then test large libraries design ideas. Okay, you can test library design ideas and then also to identify most promising scaffolds and arrow groups prior to synthesis. You can identify them using, your feature-based drug design. So I think that is my conclusion. And so I want to thank you very much for listening. Uh, I'll wait for your comments and your 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 contributions. Thank you very much. I'm sorry for taking this much time, please.
We are so much blessed. We are so much blessed. Can we react to Doc? He has delivered more than we even expected. We are so much blessed. We are so much blessed. Wow. On behalf of the Institute, Doc, we say thank you.
Thank you, sir.
Thank you. Thank you so much. Okay, we are going to react. I'm seeing some hands up already. Oh, we'll take questions and answers at this point. I believe when the lecture is well delivered, you don't even need to ask questions because you clearly understand everything. So, but still, some persons will like to ask one or two. All right. So, can I see your hands up? I'm seeing Timmy Tope. Yeah, Timmy Tope, is your hand up? I'm very sure there will not be question in this session because it was well delivered. Okay, comments and appreciation. You are free. You are free now because I know there is no question. So, comments, suggestions, and appreciation. Okay, Abiodun.
Yes, sir. Good evening,
Yeah,
everyone.
yeah.
Can you hear me, sir?
Yes, we can.
I can hear
Yeah.
you.
Yeah. Thank you, Doctor. We are so grateful, sir. May God continue to bless you and bless your ways in life, sir. In fact, the presentation was so interesting because I've been cracking my brain. I didn't really have a kind of deep knowledge on QSAR, but based on this presentation, I can see a kind of signal and better understanding because I've been comparing pharmacophore features and QSAR, but now I can see better understanding on it. And there used to be a kind of element of doubt because I normally use Schrodinger for docking and Pyrex. But there was a time I had a friend, we were trying to publish a paper, and I used Schrodinger for the docking. Then I ran the normal, the admit, I checked the normal docking, the binding affinity, and I also did MMGBSA. Then they sent the sample for molecular dynamic simulation. Then when the result came, I checked the results. Then they also ran MMGBSA, and I tried to read some papers to compare that there would be MMGBSA before the MD simulation. They also do MMGBSA just to confirm if there is no difference between the first and the last MMGBSA, just to compare. Now I now realize the importance of MD simulation, that it will give us insight into the stability and the flexibility of the complex, just to know more that okay, this particular ligand, probably the lead compound, is actually fit and stabilized after the docking. So I want to also use this period to talk to Dr. Henry that will be expecting a training on QSAR and machine learning. Thank you very much, sir. We're so grateful, sir.
Yeah, thank you very much. One of the training, somebody asked a question on MD simulation, and Dr. Siago was, replied it and also made contribution because MD simulation really, when you really want to publish a work in computational studies and then in some very strong journals, when I mean strong journals, high impact factor journals, if without MD simulation, it's not going to scale you. So it's very important. The only thing is that it can be expensive. It depends maybe on the area where somebody will run it for you. And then you may also send it overseas, if you have to, if you have the connections to do that. So that
Yes,
takes
yes.
some time, and then it's also very expensive, but it's a requirement. It's required in very, if you really want to drop your paper, I mean, in strong impact factor journals, you need MD simulation. Thank you.
Thank you, sir.
Thank you very much. Salusi, I can see your signal.
Abiudu Musulidin, something like that. Mr. Or Dr. Abiodun, please. And then, Robodo Joseph. So, Mr. Abiodun or Dr. Abiodun, please, you have the floor. Yeah, you have the floor, please. Okay, this is Imrana Salisu. Okay, you have the floor, please.
Yep. Yeah. Yes. Thank you so much, sir, for your wonderful presentation, and I really appreciate for this presentation. So I have a question, particularly on the application of the future-based drug design with regards to environmental science study. It seems like I don't know if it was network that took me out. I don't know if you talk about little bit the application of future-based drug design in the environmental science. Uh,
No, I didn't really speak much about that. Maybe Dr. Shago, if you can help us on in that, please.
All right. Thank you very much. Mr. Salusi, can you hear me if you can? Hello, Salusi, can you hear me? Okay, quickly. When we talk about future-based drug design in the area of the environment, is a novel area because we already know that these tools are applied mostly in drug design and biomedical science. But we'll now come to the aspects of the environment. We can now design molecule with certain features. That is the idea. Now, there are some environmental friendly molecules you can now design obeying certain features. That is the feature based. Like if you want to build a pesticide now, if you want to build a pesticide, so you obey the rule, the feature based. So there are some features that pesticide must possess. So you don't need to be doing much of a trial and error. Now, if you want to build an absorbent, if you want to create an absorbent, a substance that will help in removing environmental pollutants, maybe from wastewater, then there are certain features that that absorbent will possess. That is the idea of feature-based. So you are not just going to build any kind of material of your choice, but you look at the features that are necessary for absorption, So if you want to build an absorbent. Now, if you want to build a coagulant, if you want to build a coagulator, something that will help in coagulating pollutants in water cleaning. So there are certain features that coagulant must possess. That is the idea of feature-based. So we are putting up actually a training for all this because we have been discussing feature-based, pharmacophore. So we are putting up a training by March. Yes, we have already announced that by March we have a lot of 1 month to deal with feature-based drug design and also in the environmental area. So Salisi, I believe I've done little justice to that question, right?
Yeah, thank you so much, sir.
All right, thank you. Thank you.
Dr. Harry, thank you very much. There is, I think, 2 hands are up. Joseph, Roboto, and two 20, something like that, please. Joseph, please, you have the floor.
Thank you, sir. Are you hearing me, sir?
Yes, I'm hearing you, Mr. Joseph.
We are grateful for the lecture delivered.
Thank
More
you,
grace to your elbow in Jesus' name. Actually, I've been wondering about this MD simulation of 18 and concerning what you made mention of, that some of our systems that we have here may not be able to do them. What special system do you think we can have so that we'll be able to run such activity on our project? That's number one. Then number two.
Okay. 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 You know, there are some individuals really, and some schools also that have what we call a PlayStation, you know, and then of course these are computers that have very big memories, okay, and the functionality also are very powerful that can calculate, this can carry out calculations, in real time and reduce time also. Like I said, I know a lab in in uh, in the West that they do that. Also, I was in conversation with Professor Uzochukwu from Nnamdi Azikiwe, also was actually telling me how we can, establish, have, some computers to carry MD simulation in our school. And maybe I may just let this out. We are actually currently building a bioinformatics lab in our department at Uniben, and we intend to get some of these computers that we can use for MD simulation. And hopefully by next month, the lab should be up and running that we can do MD simulation. But most of the computers we have now, the 4 gig RAM and all those stuff, they may not be able to carry out real time and this simulation. So you need computers with very massive memory, and then also we are talking of terabytes, you know, uh, 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 you know, um how do you call it, motherboards to be able to carry out any simulation. If you don't have such, very, very powerful RAM in your laptop, you won't be able to do it. And that is why mostly in the US, for instance, they have centers where they have, like the center I worked in the US, University of Mississippi, they have, supercomputer they can use for MD simulation. So that's, but if you don't have supercomputer, you can carry out MD simulation, and you have a source of power that you can run it for maybe a week or two, thereabout, depending on the number of nanoseconds you want to run your simulation. So maybe after this we can speak privately if you want to, and then we can discuss more about that. So, okay.
Okay, thank you very much, sir.
Yeah, thank
My
you very much.
second question is on induced fit docking, because as
Okay.
you have what is the place of induced fit docking
Hmm.
in molecular docking? Because from your explanation now, induced fit docking and molecular MD simulation, they are almost going the same direction. Like some papers, if we can include the induced fit docking, brushing aside the MD simulation, will it be acceptable?
If I may answer your question correctly, uh, uh, induced feed docking generally, it depends really on the how do you call that, on the journal. Okay, if they can accept induced feed docking in place of MD simulation, you know but in induced feed docking really is a computational method also in drug discovery, of course, that. And it plays the role of the flexibility of the protein and your ligand to be able to stay long enough, and to elicit its its activity. In MD simulation, you are actually dealing with a flexible protein, okay, very flexible. But in your induced fit, okay, the protein is semi-flexible. Okay, it's
Okay.
semi-flexible. That's the major differences between uh, the two activities. Okay, the MD simulation, you have a flexible protein because you are trying to simulate the natural environment of the protein in the human body, or in the animal, in the organism. But in the induced fit, the protein is not completely flexible, but is semi-flexible. So those are the difference. So if for its acceptance by journals for publication, it will depend on the journal. How do you call it? The
Yeah,
journal.
standard.
Yes, focus and standard. Some journals may not accept it, while others will accept it. In fact, most journals that I know, if you don't have MD simulation, they reject your paper right away.
All right, thank you very much, sir. I have two requests.
Okay.
One of the requests is that we would like you release your contact, number one. Then along the way, the slides did not show. I couldn't see many slides that you presented.
Oh,
So
I'm sorry for that.
the slide and the contact, and the
Okay.
contact for
Okay,
further.
for the contact, you may have to consult. In fact, for the slides, I'm going to give the slides to Dr. Siago and also my contact. So you may need to consult him or contact him to give you those things, please.
All
Thank
right,
you very
sir.
much. Yeah.
Okay.
Then
Thank you, sir.
uh, Dr. Jude Twei, something like that. I think we'll share.
Hello, sir. Good evening, sir. I'm not a doctor
Okay.
yet.
Okay. Yeah. Sorry.
Okay, good evening, sir. I am so grateful for this platform. I'm very grateful. I'm of molecular biology background, so I'm just like learning molecular docking and computational chemistry through this platform. So
Okay.
most of the assignments that we are given when a training is done, I could not do it. I don't have a system that is proper for such handwork. So you did mention, sir, Dr. Vincent, that
Yes.
there's a lab in the West here that
Yes.
have all those kind of heavy systems that one could
Yes.
work with. Because
Yes.
I'm really not happy that I go through training that Dr. Henry gives and I cannot
Mm,
do one assignment. It's not I'm not happy with it. So I don't know if you
mm.
could shed more light about getting to such organizations and how to use their system, even
Okay.
if it's not for assignments, but just to practice, so that what
Mm,
Dr. Henry comes and train us, and to God be the glory, the training are even free. So I just
mm,
take notes. So
mm,
so one can just practice and one will not forget. So if one
mm.
just have such opportunity to get to such organizations. Thank you very
Yeah.
much for the training,
Yeah.
sir. And this web.
Yeah. Yeah, I think you don't really have an issue because all the training that Dr. Shago is giving, you can use your regular laptop to run all the to do all the experiment. I mean, all the assignments. Your regular laptop. Maybe if it's a 4 gig RAM laptop, can maybe core i3, core i5, core i7 laptop can do some of these assignments. What you're just basically doing is to just harvest, ligands from your databases. It hasn't given us any strong, assignment that you may need to go out of your territory to go and do. Majorly, when I say you have to go out, is especially when you are carrying out MD simulation. Your laptop may not be able to do it. Dr. Sagu told us that his lab. In fact, I've not used my laptop to do anything like that before. I have to send my work out for such calculations because I know that it's going to crash my laptop. So I don't even use my laptop. Dr. Sagu told us the other day that his laptop had been crashed almost 4 times. Trying to do MD simulation. It's not that you cannot do it with your laptop, but is your laptop strong enough? Does it have the capacity to withstand those long hours of, calculations that you require for MD simulation? So currently, as it is, during this course of the training, I think your laptop, I don't know the type you have, but if it is a Core i3, Core i4, 5, 7, Core I, any of those ones can actually do all these things we are doing. You can use those ones for your docking if you are using Pyrex or you are using, Maestro, Frontiga, all those stuff. You can just do all those docking with your regular laptop. Yes.
Oh, thank you very much, sir. Unfortunately, I don't have any Core high laptop. I have just an Intel, but thank
No,
you
no,
so much for that.
Intel these days doesn't you those things they don't use Intel these days. Anyway, however, but there are also organizations that give free laptops. You
Wow.
understand? Yes, there are organizations that
Okay.
give free laptops. So you may need to find out. Maybe you can reach me, through Dr. Osiago. Maybe we'll talk about that some maybe later.
Okay, sir. Okay,
Yeah.
sir. Thank you so
And
much. Thank you
I
very
need
much.
you.
All right. We have three more persons,
okay.
and we are going to see how fast we can go. I'm seeing
Yes,
three more
because
hands up. Ayofe, Rotimi, and Yusuf. So,
yes.
John. Okay. Uh, Ayofe, please just go ahead and make it precise.
Okay, good evening, sir.
Yeah, good
Yes,
evening.
good evening.
Okay. I don't know. Are you?
Yep. Maybe Rotimi, if you are ready, please, you can ask
Yeah,
your
good
question.
evening, distinguished lecturer. Thank you for the lecture. My questions are two, and I'll be very quick about it. One, you mentioned that you are going to have a bioinformatics lab in action very soon in Uniben.
Yes,
I was going to
very
ask.
soon. Yes.
I was going to ask that apart from your power computers, will you also consider having a sequencing facility? Maybe you can give us some relief in terms of cost, if yours will be more affordable than the private ones that we have in the country. Some of us are doing sequencing, and this is limiting our work, a lot. That's
Yes.
one. Second question
Mhm.
is that since we are already developing a bioinformatics lab, I'm suggesting that maybe you should also upscale it to
Mm.
a bioinformatics course. Maybe
Okay.
your university is going to be the first in the country that have a training platform for in bioinformatics, and that will also be a very good one for most of us, that are into bioinformatics that we cannot get certified. That is actually limiting our exposure to the outside world. Thank you.
Thank you very much. To just answer that, I don't know whether this is a very good platform to do that, but one of our professor is actually on this platform. Is currently here. Is listening to us. Through his influence in the college, what we call the postgraduate college, we're able to actually expand, the curriculum of from school chemistry. And so now we have options. And one of the options we have is bioinformatics. Okay. And I can tell you in this last, intake, we have a student currently enrolled into the bioinformatics, program in the Department of Pharmaceutical Chemistry, University of Benin. So a course is already there running, as it were. I think the section for this new intake I think it will commence on the 16th of February. I can't say, but that is about that. So yes, we may not be able to do sequencing, but if you have work related, we have sequence labs in Uniben that you can do sequencing. Yes, there are labs in Uniben that you can actually carry out your sequencing with, not too expensive. Uh, so maybe we can, contact on that now. Maybe we can talk about that off of this platform. So those are the things I can say about that. Yes, our the goal of our bioinformatics lab is that everything that you require for running a full master's program in bioinformatics will be provided by the grace of God. Yes. Well, the mechanism is in place. We are reaching out to people, to help us do all that. Yes. So I think another person, is it Dr. Hello, Dr. Tosago, are you there?
Yes, yes.
Who was the third
Uh,
person, please? The third person.
Adamu. Yeah, Professor Dr. Adamu. Are
Okay.
you there? You can
Okay.
draw
Yes.
this evening.
Good evening, everyone.
Yes. Yes. Okay. Good evening.
Um, without wasting much of our thank you very much. Let me start by thanking the presenter for the wonderful presentation. My question is, can the presenter please suggest some recommended computational chemistry textbooks that can guide young researchers and early career professionals? Thank you so much.
Yeah, thank you. Well, graciously, Dr. Siago has even given us some very, very, powerful computational textbooks that free of charge, you know. And some of these you can actually download them from a PDF drive. Some of these ones you can download them from PDF drive. They are in the public domain. And so if you go back to the test that he shared with us, some of the ones that they are just very powerful. You just need to that. So I don't think there's anyone I can recommend for the now, but the test that he gave to us, those John textbooks that he gave to us, they are very, very good. And if you harness them, you will do very well. Thank you very much. So, Dr. Shago, please, maybe you can bring this to an end or something.
Yes, because tomorrow we need to prepare for service. I think there is no more hands again. So quickly, I want to appreciate every one of you who have. John, do you have a question? Okay, maybe further questions should be dropped in our platforms.
Hello?
I believe
Hello, sir.
Yes.
Yes, sir. Sir, can you hear me?
Yeah, okay, yes, Lucy, I can hear you.
Hello?
Yes, we can hear you.
Okay. Yeah. Okay, sir. Sir, you made mention of MD simulation is meant to study
Okay.
the flexibility and the stability of ligand.
Okay.
But all like important. But sir, I have been using Schrodinger molecular difference software, and during that dosing, it has an option of selecting flexible dosing. So what does that mean?
Okay. Well, can you come over that again, please?
Okay, sir. I said, sir, I have been using Schrodinger Mistral software for molecular data, and it has option of choosing flexible dosing. So what does that mean?
But did you deploy that for MD simulation or just your normal docking? Hello, Mr. Salisu, did you use that for MD simulation? Hello?
Yes,
Hello? I can't hear you. Hello? Well, Dr. Siagu, are you there, please? Hello? Dr. Siagu, are you there? Dr. Siagu, are you there? Hello? Anybody in the house?
Hello, sir.
Yes,
Maybe it's network.
sir.
Maybe something happened.
yes, we can hear you.
We can hear you, but
Uh,
I don't know. Maybe it's
We are hearing you.
I didn't hear Mr. Salisu very well, so but I was trying to ask him a question if he used that for MD simulation, the regular normal docking. I didn't get a response from him. Anyway, but is Dr.
Yes,
Sago on the platform?
yes, can you hear me?
Hello? Yeah, I can hear you, please. Yes.
Okay. I want to reply to Salusi if he can hear me that irrespective of using a flexible docking, those are just types of docking: flexible, semi-flexible, rigid. It does not have business with MD. It cannot replace MD. Yes, it does not replace MD. So if you have option of choosing flexible docking, fine, but it doesn't have that capability of replacing MD. You must still do your simulation. Thank you.
Okay, thank you very much for that response. And maybe at this time you can close
So
the.
Hello?
I
Hello?
want to bring this session to
Hello?
yes,
Hello, Dr. Henry.
yes,
Hello? The question of that
yeah, I can hear
individual
you.
is that he said he has been making use of Schrodinger method.
Yes.
That software has an option for MDs and that can he make use of
Okay.
No, the
If if the software has
that
option
option
for
there.
MD
So the
simulation,
doctor
let
was
him
trying to
try it.
ask him whether he used it for MD or document.
Uh, if
Uh,
if Master has option for MD, let him try it, but you should be careful. Yes, that is my opinion. Uh, since it has option
all
for
right.
him, let him try it, but you should be careful. Yeah.
There is no option for MD
and probably
simulation on Code Danger. I'm using it too.
All right. Once again,
Okay.
once
All
again,
right. Thank you.
I want to say
You can also run
a
MD
very big
simulation.
thank you
You can also run MD
to
simulation.
everyone who are privileged to be here. Quickly, thank you for being part of Hendeb. We are just trying our best to see that we bridge the gap because we have a lot of gap. So we bridge the gap. And I want to say that Uniben is really growing massively strong. And I think Hendeb, after now, we're going to have a, a background discussion with Uniben so we can also collaborate with them in some of the work they are doing. So once again, thank you. Next week, we have a training. Next week is a free training. You can join. Is on molecular modeling. So I think that is the training. If you can see my screen, can you see my screen? So next week is molecular modeling from 21st to 22nd. You can avail yourself the opportunity, and then we are coming up by February. We have a training, 1 month training on green chemistry by February. You can avail yourself that opportunity. And also we have a beginner's course for those who are interested in having a foundation of computational chemistry. You can enroll in this training. It's a beginner's course. It's starting on the 28th of January. So a lot of training has been on the pipeline. A lot of training is already on the pipeline. We are encouraging you participate in our free training, participate in the paid training, and I believe that it's worth it. By February, this is our calendar for the month. We have already exhausted that of January. Yeah, we just exhausted that of January. Now for February, I want you to keep your mind receptive because from the first week of February, we'll be launching a free training on chemoinformatics across life sciences. While on the second week of February, we are dealing on predictive modeling in biomedicine. Then, okay, that's our webinar for February. It will be on predictive modeling in biomedicine. Our second free training is QSAR. So we have been hearing about QSAR. We have a training for that in February, a free training for QSR and QSPR. So avail yourself these opportunities every month in the year. Throughout this year is training. So I believe every week every week is back to back, back to back, back to back. We are all trying our best to see that we bridge the gap. Yes, we want to bridge the gap. And once more, thank you. At this point, I would say good night. And a very big thank you to our facilitator. We owe you a lot. God bless you. To Uniben, we say thank you. Yeah,
Please,
thank you
God bless
very
you.
much.
remember to give
Thank
us
you
the
very much.
slide.
God bless.
Thank you.
Thank you very much.
Yes,
Good
yes. We shall send
night.
the slides and the video recording by tomorrow. So you can use that and go to whatever we have discussed. And then for those who want to reach out to Doc, probably you can slide my DM. Then I'll release his possible contact to his permission. Once again, thank you. God bless you all. Good night.
Good night.
Good night, sir.
Good night, sir.
Good night, sir.
Night. Thank you so
Yeah,
much.
yeah, thank you.
Good night, everyone.
Good night, sir. Thank you very much. Good night.
Thank you. Professor Adiola, I can cite you. Good night.
Thank you, sir. Good night, sir.
Yeah, yeah, good night.
Thank you, sir. Good night, sir.
Azoka Ogechi, my friend. Good night, everyone.
Thank you, Dr. Henry. Good night, too. Thank you.
Thank you very much, sir. Good night.
Good night. Thank
Thank
you.
you.
Good night, sir.
Good night, sir.
Oh, I define